There is an organ inside your body that your doctor almost never mentions. It is not your heart. It is not your brain. It does not have a dramatic name that appears in headlines or health campaigns. Most people could not point to it on a diagram. And yet right now, at this exact moment, it is regulating your immune system, producing compounds that affect your mood, controlling the rate at which you absorb nutrients, communicating directly with your brain through a dedicated neural highway, and determining whether the inflammation running through your body is being contained or amplified. It weighs approximately 1.5 kilograms. It stretches nearly 9 meters if uncoiled. And inside it lives an ecosystem of approximately 38 trillion microorganisms, bacteria, viruses, fungi, and archaea, that outnumber your own human cells and collectively carry 150 times more genetic material than your entire human genome. We are talking about the gut, not the simplified version you learned about in school, a tube that digests food and eliminates waste, the real gut. The one that researchers have spent the last 2 decades discovering is arguably the most influential organ system in the human body, connected to conditions as seemingly unrelated as depression, autoimmune disease, cardiovascular risk, obesity, skin disorders, and cognitive decline. In this video, we are going to go inside that system layer by layer and show you exactly how it controls far more of your health than you were ever told. Section 1. What the gut actually is. Let's begin at the foundation, because the gut most people picture and the gut that science has revealed are almost completely different structures. The gastrointestinal tract begins at the mouth and ends at the rectum, a continuous tube running through the center of the body that is functionally divided into specialized regions, each with a distinct architecture and biological role. The stomach breaks food down chemically and mechanically. The small intestine, which despite its name is the longest section at approximately 6 meters, is where the vast majority of nutrient absorption occurs. The large intestine, or colon, handles water reabsorption, electrolyte balance, and houses the densest concentration of the gut microbiome. But the most important structural feature of the gut is one that is invisible to the naked eye. The inner surface of the small intestine is lined with millions of tiny, finger-like projections called villi, and each villus is covered with even tinier projections called microvilli, together forming a structure called the brush border. This architecture increases the absorptive surface area of the small intestine to approximately 250 square meters, roughly the size of a tennis court, packed into a tube that fits inside your abdomen. Beneath this absorptive surface lies one of the most significant biological structures in the body, the enteric nervous system. This is an independent neural network embedded in the gut wall, containing approximately 500 million neurons more than the spinal cord. It operates autonomously, regulating gut motility, secretion, and blood flow, without requiring instruction from the brain. Researchers now refer to it as the second brain, not metaphorically but structurally, because it shares the same neurotransmitters, the same signaling mechanisms, and the same developmental origins as the central nervous system. Section 2. The ecosystem running the operation. Now let's go deeper into the living community that modern science has identified as the gut's most powerful operating system. The gut microbiome is the collective name for the trillions of microorganisms living primarily in the large intestine. For most of medical history, these organisms were viewed as passive passengers, harmless commensals hitching a ride through the digestive tract. What research over the past 2 decades has revealed is something entirely different. These organisms are active participants in nearly every major biological process in the human body. They produce vitamins including a significant portion of your vitamin K and several B vitamins that the human body cannot synthesize independently. They ferment dietary fiber into short-chain fatty acids, particularly butyrate, propionate, and acetate compounds that serve as the primary energy source for the cells lining your colon, regulate immune function throughout the body, influence insulin sensitivity, and cross the blood-brain barrier to affect brain chemistry directly. The microbiome collectively produces more than 30 neurotransmitters and neuroactive compounds, including 95% of the body's total serotonin, which is produced in the gut, not the brain. Serotonin in the gut regulates intestinal motility and communicates with the enteric and central nervous systems. The gut-brain connection runs in both directions through the vagus nerve, a direct communication highway connecting the gut and the brain, and through systemic circulation of microbial metabolites that reach brain tissue and influence neurotransmitter availability, neuroinflammation, and mood regulation. A diverse, balanced microbiome produces anti-inflammatory compounds, competes with pathogenic bacteria for resources, trains the immune system to distinguish friend from foe, and maintains the integrity of the gut barrier. A disrupted microbiome, reduced in diversity and dominated by fewer species, does the opposite across all of those functions simultaneously. Section 3. Where 70% of your immunity lives. Let's address something that most people find genuinely surprising when they first encounter it. Approximately 70% of the entire immune system is located in and around the gut. This is not a coincidence. It is a direct consequence of the gut's function. The intestine is the largest surface of contact between the inside of your body and the external world. Every meal you eat introduces thousands of foreign proteins, bacteria, fungi, and chemical compounds into direct contact with the intestinal lining. The immune system had to evolve to be present at this interface in enormous force, to continuously distinguish between what is food, what is beneficial microbial life, and what is a genuine threat requiring a defensive response. The gut-associated lymphoid tissue, the collective name for the immune structures embedded throughout the intestinal wall, contains more immune cells than any other organ in the body. These cells are in continuous dialogue with the microbiome. Beneficial bacteria actively educate immune cells, training them toward tolerance of harmless substances and away from unnecessary inflammatory responses. This education process begins at birth. Research shows that the first microbial colonization of the infant gut, in the days and weeks after birth, programs immune behavior patterns that persist for decades. When the microbiome is disrupted, through antibiotic use, chronic stress, ultra-processed diets low in fiber, or other factors that reduce microbial diversity, this immune education fails. The result is an immune system that is poorly calibrated, more prone to inflammatory overreaction, more likely to develop sensitivities to food proteins, and more susceptible to the immune dysregulation that underlies autoimmune conditions. Research now links gut microbiome disruption to a growing list of autoimmune and inflammatory conditions including rheumatoid arthritis, inflammatory bowel disease, type 1 diabetes, multiple sclerosis, and psoriasis. Conditions that appear to have nothing to do with digestion but share a disrupted gut immune interface as a common upstream factor. Section 4. How your gut controls your mood. Here is one of the most consequential scientific developments of the past decade, and one that has not yet reached mainstream health conversations in any meaningful way. Your gut and your brain are in constant bidirectional communication through a system researchers call the gut-brain axis. This system operates through three parallel channels simultaneously. The first is the vagus nerve, the longest cranial nerve in the body, running from the brain stem all the way down to the abdominal organs, which carries signals from the gut to the brain at a ratio of approximately 9:1, meaning for every one signal the brain sends down to the gut sends 9 signals up to the brain. The second channel is the endocrine system. Gut cells produce over 20 distinct hormones that enter the bloodstream and affect brain function, appetite regulation, stress response, and energy metabolism. The third channel is the immune inflammatory pathway. When the gut microbiome produces inflammatory compounds, those compounds reach the brain through the bloodstream and directly affect neuroinflammation, which research increasingly links to depression, anxiety, and cognitive decline. The serotonin connection deserves specific attention. 95% of the body's serotonin is produced by specialized cells in the gut lining in response to signals from gut bacteria. This gut-produced serotonin does not cross the blood-brain barrier directly, but it regulates the enteric nervous system and communicates with the brain through the vagus nerve in ways that influence mood, anxiety, and stress reactivity. Research shows that germ-free animals, those raised without any gut microbiome, show dramatically abnormal stress hormone responses and anxiety-like behaviors that are partially corrected when specific bacterial strains are reintroduced. Studies in humans show that people with lower gut microbiome diversity show higher rates of depression and anxiety, and that certain probiotic interventions produce measurable improvements in mood and stress reactivity, an effect now described as the psychobiotic response.
Section 5. Leaky gut, the barrier that breaks down. Let's address one of the most discussed and most misunderstood concepts in gut health: intestinal permeability, commonly referred to as leaky gut, and explain what it actually is at the biological level, separate from the oversimplified versions that circulate in popular health content. The intestinal lining is a single cell layer thick. That is one cell standing between the contents of your gut, partially digested food, bacteria, bacterial fragments, and metabolic byproducts, and your bloodstream. What makes this barrier functional is not its thickness, but the tight junctions between those cells. Protein structures called claudins, occludins, and zonulins that lock neighboring cells together and prevent anything from passing between them. Only what is actively transported through the cells themselves, selected nutrients, absorbed through specific receptor-mediated processes, should enter the bloodstream. When tight junctions are disrupted, the barrier becomes selectively permeable in ways it should not be. Bacterial fragments called lipopolysaccharides, components of gram-negative bacterial cell walls, can pass through into circulation. Partially digested food proteins that the immune system has never encountered in the bloodstream before can enter and trigger immune responses. The result is a state of chronic low-grade systemic inflammation driven by constant low-level exposure to substances that belong inside the gut but not in the bloodstream. Research shows that tight junction disruption is caused and perpetuated by several well-established factors. A diet low in fiber reduces butyrate production, and butyrate is the primary fuel for the intestinal epithelial cells that maintain junction integrity. Chronic psychological stress increases intestinal permeability through cortisol-mediated signaling. Alcohol directly disrupts tight junction proteins. Certain medications, including non-steroidal anti-inflammatory drugs, reduce the protective mucus layer that shields the intestinal lining. Elevated intestinal permeability has been documented in patients with inflammatory bowel disease, celiac disease, type 1 diabetes, obesity, non-alcoholic fatty liver disease, and depression, not as a consequence in all cases, but as a contributing upstream factor. Section 6. What destroys the gut, and what science says rebuilds it. Let's be direct about the specific factors that damage gut function. because understanding the mechanism makes the prevention logic obvious rather than arbitrary. Antibiotics are the most powerful single disruptor of the gut microbiome available. They are also medically essential and genuinely life-saving when used correctly. The problem is the collateral damage. A broad-spectrum antibiotic course that eliminates a targeted pathogen also eliminates hundreds of commensal bacterial species simultaneously. Research shows that a single course of antibiotics can reduce microbiome diversity by 30% or more, and that in some individuals, recovery to baseline diversity takes 6 months to 2 years. Repeated courses compound this effect, and the cumulative disruption of multiple antibiotic courses across a lifetime has been associated with increased risk of inflammatory bowel disease, obesity, and metabolic dysfunction. Ultra-processed foods, defined as industrially manufactured products containing ingredients that do not exist in home kitchens, including emulsifiers, artificial sweeteners, modified starches, and synthetic flavor compounds, have shown consistent associations with reduced microbiome diversity in large population studies. Emulsifiers, in particular, appear to physically disrupt the mucus layer protecting the intestinal lining, bringing bacteria into closer contact with epithelial cells than they should be. Dietary fiber is the primary fuel for beneficial gut bacteria. Without it, the species that produce butyrate and short-chain fatty acids decline rapidly. Research shows that microbiome diversity responds to dietary change within 72 hours, meaning the microbiome is sensitive and responsive to what you eat with remarkable speed. What rebuilds the gut is less dramatic but well supported. Dietary diversity, eating a wide variety of plant foods, is the most consistently evidence-backed intervention for microbiome diversity. Fermented foods containing live cultures, including yogurt, kefir, kimchi, and sauerkraut, have shown measurable microbiome benefits in controlled studies. Adequate sleep, regular physical activity, and stress reduction all improve microbiome composition through mechanisms involving cortisol, circadian rhythm, and gut motility. Section 7. The gut and disease. The connections medicine is still catching up to. Let's look at the broader picture. Because the gut's influence on systemic health extends into territory that continues to surprise researchers and that has not yet been fully integrated into standard medical practice. The connection between the gut microbiome and cardiovascular disease is one of the most significant recent findings in the field. Certain gut bacteria metabolize a compound called choline, found in red meat and eggs, into a substance called trimethylamine, which the liver then converts into TMAO, trimethylamine N-oxide. Elevated TMAO levels in the bloodstream are independently associated with increased cardiovascular risk, atherosclerosis, the buildup of plaque in artery walls, and adverse cardiac events. The key insight is that the same dietary input produces different TMAO levels in different people depending on the composition of their gut microbiome, meaning cardiovascular risk from certain foods is partly determined by who is living in your gut. The gut-cancer relationship is emerging as significant across multiple cancer types. Research shows that the microbiome influences cancer risk through immune regulation, production of carcinogenic or anti-carcinogenic compounds, and modulation of systemic inflammation. Specific bacterial imbalances have been associated with colorectal cancer risk, and the microbiome's response to chemotherapy and immunotherapy appears to influence treatment outcomes in ways that researchers are actively working to leverage therapeutically. The gut-metabolic connection is perhaps the most established. Gut microbiome composition influences insulin sensitivity, weight regulation, and fat storage through multiple mechanisms, including short-chain fatty acid signaling, bile acid metabolism, and direct influence on appetite-regulating hormones. Research shows that germ-free mice are resistant to diet-induced obesity, and that transferring microbiome samples from obese animals to lean germ-free animals transfers the metabolic phenotype along with the bacteria. Section 8. Reading your gut. The signals worth paying attention to. Let's bring everything together with the most practical lens, because what you now understand about the gut changes how you should read the signals your body sends every day. Chronic bloating, irregular bowel habits, unexplained fatigue, recurring mood disruption, skin conditions, including acne and eczema, frequent infections, and food sensitivities that seem to be multiplying over time are not separate, unrelated complaints. They are often overlapping symptoms of a gut ecosystem under chronic stress, and they deserve to be understood as a system rather than treated as isolated problems. The gut gives you daily feedback through bowel habit consistency, stool characteristics, gas production, and abdominal comfort. Research supports the Bristol Stool Scale, a clinical tool that classifies stool consistency from type 1 to 7 as a reliable indicator of gut transit time and hydration status. Types 3 and 4, smooth and sausage-shaped, represent optimal transit. Consistently hard pellet stools or loose urgent movements both reflect gut function that is outside the optimal range and worth addressing rather than normalizing. Persistent symptoms that include blood in the stool, unexplained significant weight loss, nocturnal symptoms that wake you from sleep, or a family history of colorectal cancer warrant colonoscopic evaluation. These are not anxiety-inducing possibilities to dwell on. They are simply the signals the gut sends when something beyond lifestyle optimization requires clinical attention. The deeper takeaway from everything covered in this video is this: the gut is not a passive digestive tube. It is an active regulatory organ communicating with your brain, educating your immune system, influencing your metabolism, and shaping your risk for conditions that seem to have nothing to do with digestion. Taking it seriously, feeding it diversity, protecting its barrier, preserving its microbial community, is not a wellness trend. It is one of the most evidence-backed investments you can make in your long-term health. The organ that controls everything has been with you your entire life. Now, you know what it actually does.